Hi everyone, today I would like to continue the next part in the sexual reproduction, that is the 25th part, very essential for the competitive examinations at the same time board examinations. It's nothing but pollen pistil interaction. Just moving on to the topic. See this, sexual reproduction in flowering plants, part 25. Subtopic is pollen pistil interaction. Then here, directly one statement has given, pollen does not guarantee the transfer of the right type of pollen. If any pollen reaches to the stigma, then immediately it won't give. It may give, may not give. That's what may not give any kind of guarantee for the germination of the pollen. Right? Now here, during transfer of pollen, that means pollination, the pollen grain or pollen can be divided into two types. After reaching the pollen grain on the stigma, then whether it is the following in the further functions, further events or not, based on that pollen are divided into the two types. What are those mean? Right pollen, wrong pollen. Two types are right pollen, wrong pollen. Now here right pollen is otherwise called as compatible pollen. Wrong pollen is nothing but incompatible pollen. Now in our syllabus we need to talk about the right pollen much, and there is a one identification is there on the wrong pollen. First let me complete with the wrong pollen. What is the wrong pollen? Now here I'll tell you, in one particular area, three plants are there. Three plants are having flowers, they belong to flowering plants. First datura central it is present, one side mango, other side is hibiscus. These all are having the stigma, anther also. Now here, mango flower, anther is released the pollen and reached onto the datura. Then the stigma of the datura is not accepted. Then it is called incompatible. Other species. Hibiscus also same thing. Hibiscus pollen reached to the stigma of the datura. Then also datura stigma is not accepted. That is also incompatible. Then this is the incompatibility, mango and hibiscus. Now if you see datura plant itself, the anthers of the same flower produces the pollen that reaches to the stigma. Then the same flower stigma is not accepted for the further events. Then it is called self-incompatibility. Self-incompatibility. Self-incompatible. It is for the evolution purpose. It will be helpful for the evolution of this species. Next here, regarding to this, what are the points are mentioned? Often the pollen grains of the wrong type are either from the other species, mango and either from the other species, here mango, here hibiscus, or from the same plant, is nothing but incompatible. Next, if the pollen is wrong type, the pollen, the pistil rejects. The pistil is there, no stigma rejects. Pistil rejects the pollen by how? Further events are nothing but preventing the pollen germination on stigma or preventing the pollen tube growth. After germination, growth is happening in the style. That is on the stigma. It is on the stigma, germination, growth in the style. This is essential for us. Now this is the wrong type or incompatible point of view we need to discuss. Now we need to talk about much on the right type. Right pollen or incompatible. Here, if the pollen is right type, the pistil accepts, here it is rejects, accepts the pollen and promote the pollen germination on stigma, there prevents the pollen germination on stigma, and here promotes the post-pollination events, means the growth of the pollen tube in the style that leads to the fertilization. Next, further what is happening? The ability of pistil to recognize the pollen and followed by acceptance and rejection is the result of a continuous dialogue. Continuous dialogue means process or reactions between two things, pollen grain and pistil. The acceptance or further events of the pollination, that means pollen tube growth on the stigma in the style, is based on this, the continuous dialogue between the pollen grain and pistil. This dialogue is mediated by, very important equation, chemical components of the pollen which interacting with the pistil. Nowadays, in the recent years, the botanists have been identified that some of the pollen pistil components and pollen pistil interactions leading to the recognition and then followed by acceptance and rejection. Next, incompatible pollination, how pollen pistil interaction is going on. Now first I would like to explain, then afterwards I would like to explain the word here. This is a pollen grain. It has exine and intine inside the cytoplasm. This is a pistil, stigma, style, ovary, and here that pollen grain, which is the right one, it is the right one. If it is the right, that is what we need to discuss here. Right pollen or compatible pollen, then immediately stigma accepts. Then this stigma, whenever the stigma accepts, then immediately the pollen tube germination happens. This is what we have given. Then after this will be germinate up to the ovule. Then after some functions are going on, that I will talk to you now. Incompatible pollination, the pollen grain germination on stigma and produce a pollen tube through one of the germ pores. So many germ pores are present here. The pollen tubes are not forming. Many pollen tubes are not forming from the one pollen grain. Only one pollen tube, that too only from one germ pore. This is an equation for you. Next, here this is the ovary. Ovary is having the ovaries. The contents of the pollen grain move into the pollen tube. Generally, whenever the pollen tube germination happens from the germ pore, that time the content, whatever it is present inside, that is also moving into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches finally the ovule. This is also how it is the tissue of the stigma, and after stigma to the style, then finally reach the ovule. Now here, as you know that pollen grains are shed down from the anthers, either 2 cell state or 3 cell state. 2 cell state means it is the 60%, 3 cell state means 40%. If it is the 2 cell state, how the 3 cells are forming? That is, we need to come to that. Now, as we know that in some plants, the pollen grains shed at 2 cell state, that is 60%. What are those 2 cells means? One is the vegetative cell, second is the generative. In these plants, the generative cell divides mitotically and forms 2 male gametes. Now here if you see, this is the pollen grain, which is the stigma, then pollen tube forms. Here, yellow color is one generative cell, one vegetative cell, red color is nothing but generative cell. Then after pollen tube is growing tissue of the stigma and tissue of the style. Then while here single red color is there, no, that is the cell, that is the generative cell. It is divided mitotically and produce the 2. That means 2 male gametes. So one generative cell produces the 2 male gametes. That is what I mentioned over here. Okay. One generative cell, that is the haploid in condition, and produces the 2 male gametes. That is also haploid in condition. This diagram is very important. This blue color is nothing but a pollen tube. Pollen tube starts from the stigma to the spike. If it is the 3-cell state, there is no problem. 3-cell state, pollen grains are there 40%. Then from the beginning itself only, that is what are the 3 cells means, one generator cell, 2 male gametes. From the beginning itself only, it is carried. Pollen tube is carried. Next, the pollen tube reaches the ovary. Then it enters into the ovule through the micropylar region. You know that micropylar region. Here if you see that, here the integuments are open. That is the micropylar region. Opposite was the chalaza region. Inside the embryo site is present. Now here what happens, you know? Here the pollen tube is growing like this and it enters the stigma to the ovary, ovary to the ovule. It is entering into the ovule. In the ovule, how it is entering? Ovule, if you see that embryo site is present centrally. In the embryo site, egg apparatus. You see that this is the egg apparatus, one synaptic this side and other side the other synaptic. The middle one egg cell is present. These 3 collectively called as egg apparatus. If you see that this synergid and this synergid are having special structures called as filiform cells. Filiform apparatus. The filiform apparatus are helpful for the guiding of the pollen tube. If you see the blue color is nothing but a pollen tube. That pollen tube is entered into the micropylar region to the embryo sac into the, these are the green color are nothing but filiform apparatus into the directly any one of the filiform apparatus of the synergid. It is not dividing into 2 parts, pollen tube. Then after finally it is releasing into this. Now, what is the journey of the pollen tube? First, pollen tube starts from the germ core to the stigma to the style to micropyle to the filiform apparatus of any one of the synergid, and finally it enters into synergid to the egg cell. This is very important question for us. Here only most of the people are getting the confusion. If you go through that, it's easy to identification of the journey of the pollen tube. Now the main function of the filiform apparatus present at micropylar part of the synergids is very important. Guiding the entry of the pollen tube into the ovule. This is a main question for us. Guiding the entry of pollen tube into the ovule. Now here, from the deposition, now here total events are present. From the beginning to end, whatever the events are there, those all are calculated with pollen pistil interaction. See, all the events from pollen deposition onto the stigma until the pollen tube entry into the ovule are together referred as pollen pistil interaction. This is what we need to understand the pollen pistil interaction. Many questions are there. Please go ahead for this. Okay. So it is very essential for all of you people. Please go through and all the best. Get good result from this area.